Hello, my name is Jordan Lamont. I'm a junior in the Department of Biological Sciences, and I began this project with my research mentor, Dr. Andrus, with the idea of choosing any gene from any organism to express and purify. So I ended up choosing Serratia marcescens because it's a bacteria commonly found in hospitals and causes nosocomial infections. It's known for its multi-drug resistance. This resistance relies on many different factors, including efflux pumps. The efflux pump that I wanted to focus on is the TetA, TetR efflux pump system that combats the antibiotic tetracycline. TetR produces the repressor protein that binds to the shared operons between the two genes, and in the presence of tetracycline, it will preferentially bind to the tetracycline and release its hold on the DNA, which will allow TetA to produce an efflux pump that removes tetracycline from the cell. So firstly, the DNA from Serratia was extracted, and then primers were designed to include restriction sites that allow for ligation into the expression vector, and PCR is performed using these primers to isolate and amplify the TetR gene. Figure 2 shows the results of the successful PCR, and it's got one band around the 650 base pair marker on the ladder. After the PCR solution is cleaned up, it was ligated into the cloning vector pGem-T-EZ and transformed into competent E. coli. The colonies containing both the plasmid and the gene were selected to grow more and digest using the sites added to the primers. Figure 3 shows a few colonies chosen and restricted using Echo R1 to validate if the colony contained both the plasmid and the gene. Only the middle one proved to have both. So this is the colony that was preserved and used in the later experiments. After this colony was proved to have both the gene and the plasmid, the restriction sites that were added to the primers were then used. And after that, the pinpoint expression vector would have been digested using the same restriction enzymes, and the gene would have been ligated into the vector to be expressed and purified using that system. However, time was limited, and I only was able to accomplish up to the restriction digest step. The restrictions were successful, but removing the band from the gel was almost excruciatingly difficult. After a few trials, my mentor and I concluded that the reagents were likely just too old, and a new kit was ordered, but there just wasn't enough time to use the new reagents before this presentation.